Hello and welcome back. Today's podcast will be all about excretory products and their elimination. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, chloride, phosphate, sulfate, etc., either by metabolic activities or by other means like excess ingestion. These substances have to be removed totally or partially. In this chapter, you will learn about the mechanism of elimination of these substances with special emphasis on common nitrogenous wastes. Ammonia, urea, and uric acid are the major form of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with a minimum loss of water. The process of excreting ammonia is ammonotelism. Many bony fishes, aquatic amphibians, and aquatic insects are ammonotelic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surface or through gill surfaces in fish as ammonium ion. Kidney do not play any significant role in its removal. Terrestrial adaptations necessitated the production of lesser toxic nitrogenous waste like urea and uric acid for conservation of water. Mammals, many terrestrial amphibians, and marine fishes mainly excrete urea and are called ureotelic animals. Ammonia, produced by the metabolism, is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, land snails, and insects excrete nitrogenous waste as uric acid in form of pellet or placed with a minimum loss of water and are called uricotelic animals. A survey of animal kingdom presents a variety of excretory structures. In most of the invertebrates, these structures are simple tubular form, whereas vertebrates have complex tubular organs called kidney. Some of these structures are mentioned here. Protonephridia or flame cells are the excretory structures in platyhelminths. Flatworms, example planaria, the excretory structures in platyhelminths. Rotifers, some annelids, and the cephalochordata, amphioxus. Protonephridia are primarily concerned with ionic and fluid volume regulation, that is osmoregulation. Nephridia are the tubular excretory structures of earthworm and other annelids. Nephridia helps to remove nitrogenous waste and maintain a fluid and ionic balance. Malpighian tubules are the excretory structure of most of the insects, including cockroaches. Malpighian tubules helps in the removal of nitrogenous waste and osmoregulation. Antennal gland or the crine gland performs the excretory functions in crustaceans like prawns.